Good morning, students. Today I am going to present a video representing the estimation of blood glucose. So, these are the reagents which I was talking about. So, there are two reagents specially for conduction of this blood sugar estimation. The first one is the alkaline copper tartrate. Then this one is the green one. This is the arsenomolybdate reagent. Okay, so I told you in the class we need to first precipitate the proteins present in the blood sample by doing a procedure for precipitation of proteins. For that precipitation we need, I told you this one, zinc sulfate, barium hydroxide and distilled water and blood sample. Actually that is the experiment we have to do for precipitation, but for the practical purpose we are giving the blood sample, so we are omitting that part. Okay, but I am just showing the what are the reagents required for the precipitation, but we are not going to do, since we had made this sample an artificial sample, not blood sample, so we omit that part. Okay, I think you already know how precipitation of proteins is done. See, we put this one, see, distilled water, this is the distilled water, then 3.5 distilled water, then 0.1 ml blood sample, then 0.2 ml of barium hydroxide and 0.2 ml of zinc sulfate. All together the total volume comes to around 4 ml. Okay, so we omit that part since we are giving an artificial blood sample. Okay, so next what we are going to do is that we now going to start the procedure. Okay, so we take 3 test tubes. One is marked as blank, it's marked as B. Okay, then we take another test tube that is marked as standard, marked as S. S means standard. Then the third test tube, which is our which is going to be the test, we mark as a T. Okay, so we take 3 test tubes, blank, standard, test. Now accordingly, provided the procedure, we are going to add this supernatant. Means what? We already assume that we have taken the supernatant part of the blood sample. Understand? So this is going to be the test, that is the supernatant part. Now supernatant part we are going to take how much? According to the procedure, in the test tube, that is T tube, we are going to take around 1 ml. Okay, 1 ml. 1 ml. Okay. Then comes the standard glucose solution. Okay, I have told you everything how to prepare a standard or working glucose solution in the lecture. Okay, I already told. I'm not going to repeat it. Okay, so standard glucose solution or working blood glucose, standard glucose is this one which I've already prepared. Okay, see, I'm not going to repeat the how to prepare and all. Okay, this working standard is being prepared from the stock standard. Okay, it is given in the lecture. Now how much we are going to take? Working standard glucose 1 ml. So we see, standard means S. Here we take 1 ml again. Now for the blank we are not going to take anything. Okay, now this one, then distilled water. distilled water. Distilled water, how much we are going to put? We are going to put 2 ml to both the 3 tubes. Okay, to both the 3 tubes. 2 ml, 2 ml, okay. 2 ml distilled water is put in the blank. Then accordingly 1 ml is put in the 1 ml of distilled water is put in the standard, okay, and 1 ml again in the test. Okay. Now after that we are going to put the alkaline, that is alkaline copper tartrate. Okay, it is in all the 3 tubes, 2 ml, 2 ml, 2 ml. Okay, then this complete our addition of various reagents in the 3 tubes. Okay, which is the volume comes around 4 ml, 4 ml, 4 ml each. Okay, now these test tubes are being taken now, and then
3.
So this is the 3 tubes blank standard test. Already added the various reagent depicted in the practical book. Okay, after that, after this addition, we are going to put in the boiling water bath for around 10 minutes. Okay.